Hi everyone, I know I'm in the way of everyone getting lunch, so please bear with me for the next 7 minutes. So as introduced, I'm Michelle. I'm currently a PhD student at Professor Craig McLean's lab here at the University of Oxford. And I'm going to tell you a bit about my PhD project, which is to use phages to combat AMR plasmids. So AMR needs no introduction in this room. And what I want to highlight is that the spread of AMR genes in bacterial pathogens are commonly driven via mobile genetic elements like plasmids. And this is because plasmids often can carry more than one AMR gene, leading to the rise of multi-drug resistant bacterial pathogens. So we thought, why not let's target these plasmids directly with phages? And phages has been kindly introduced by Melissa earlier, so I hope everyone's kind of familiar. And the class of phages that I'm working with specifically is called plasmid-dependent phages. And why they're special is because they require the presence of a conjugated plasmid, specifically its pili, in order to target and infect the bacterial host. And the model PDP that I'm working with is also known as PRD1, and it has been reported to target INCP, which is a broad host range plasmid, in a variety of bacterial species, including several critical pathogens from the Enterobacteriaceae family, like E. coli, Klebsiella, and even Pseudomonas. So these EM photos here were actually taken in a collaboration between Daniel, my postdoc who's here, and the Sanger Institute. And as you can see, let's see if this works. This is actually the pili, and these are the virions that are attached to it. And it's really exciting because this is one of the first EM photos we have that directly show the phage on the pili. So, like any good phage scientist, I started off with a screen. So I measured my phages in a bacterial killing assay against clinically relevant AMR plasmids that I have moved into lab strain NG1655. So I know most people might not have worked with phages before, so let me introduce you a standard assay that we use, which is called a bacterial killing assay or a plaque assay. So what I would do is I would drop a high titer stock of my pure phages onto a bacterial lawn. If there's a clearing that you can see here, it means that there's successful infection and killing. So having done the screen, I noted that INCP and INCW plasmids are able to be targeted by my phage. But that's great. However, I feel like I don't quite believe this standard assay that people have used, and there's more and more reports that we should look into alternative methods. So inspired by COVID, I also developed a qPCR assay that allows me to measure viral titer directly by boiling it and measuring the DNA. So, as expected, it really nicely recapitulated my data that I found earlier. So, this light blue here is at 0, and this dark blue is at 24 hours, and for all these 3 plasmids in the pink box, there's an increase in viral titer. However, what was really interesting is that I found that there's one INCX4 plasmid that carries a last resort antibiotic, colistin, and I can see that there is a significant increase in viral titer. However, I don't notice any plaquing at all. So clearly there must be some cryptic interactions happening between this plasmid specifically and my phage. However, just because replication happened doesn't tell us anything about suppressing conjugation and thus blocking the spread of AMR. Therefore, I developed a conjugation assay where I mix my donor strain with my plasmid of interest and the recipient. And the main aim is to count the number of transconjugants that receive the plasmid. So the red dots are the absence of phages, and the blue dots means I've added the phage. And really elegantly, this experiment actually shows that the presence of phage is able to suppress conjugation by almost 1 log, minimally, and in some cases, up to below detection limit. However, none of this actually explains why there's a mismatch in my data. This INCX4 plasmid clearly doesn't look like it's being killed, yet there is obvious data that shows that it's being replicated and targeted, and it's a novel target of PRD1. So I also developed a flow cytometry data experiment where I would dye my phages with a DNA binding dye and then run it through after incubating with the bacteria. This gives me almost single cell resolution of what is happening between the phage and the bacteria. So on the left, this is what happens when you have an obvious killing effect, and this is a clear plaque. It's a normal distribution, and this cartoon kind of illustrates that it means that the phages are bound to the bacteria. So when I don't see this plaque I expected kind of this to just not really be there or be much less. Instead, what I saw is a bimodal distribution. And this indicates that there must be some heterogeneous population within this culture, whereby one portion of the bacterial population is lowly bound by phage, while the other appears to be bound even higher than the one that has an obvious killing effect. So I know everyone wants to go for lunch, and I hope that everyone can remember the three main takeaways of my project thus far, is that firstly, plasmid-dependent phages can target and kill bacteria, even in the absence of a clear killing effect via the plaque assay. Secondly, PDPs are able to suppress conjugation, thereby preventing the spread of AMR plasmids, even in heterogeneous populations. Last but not least, the flow cytometry data actually highlights how these heterogeneous phage-susceptible populations might drive cryptic interactions, and this widens the field of what kind of phages we can look for and use in clinics, and hopefully someone else can take the mantle for that. So I just want to thank everyone for your time, attention, and to Daniel, my postdoc, and Craig for all their support. Thank you.
Hi everyone,